Hello, everyone. Welcome back to our Systems Biology podcast. Today, we will discuss QSP, Quantitative Systems Pharmacology. In short, it is acronym of Systems Biology for Pharmacology, the drug development. And to discuss with that, we have our guest, Dr. Gaurav. He's a veteran in this field of data analytic, model building, QSP, almost more than 1 decade's experience in the pharma and life science industry. Welcome, Gaurav, to our podcast.
Thank
Nice
you.
to
It's a
see
pleasure
you
to be here.
after a decade. Uh, it is really good to see Gaurav again. Dr. Gaurav Dwivedi. He was one of our earliest students. I think he graduated from IIT Guwahati Biotechnology Department in 2008.
That's right.
Then Gaurav, you moved to Georgia Tech, right? Georgia
That's
Tech to do PhD in bioengineering, and eventually started your career in the life science industry. And for almost 15 years, you have worked in the field of data analytic in pharmacology, making models, QSP. And Gaurav is currently in a very senior position. He is the director QSP in Mark. So again, once again, welcome Gaurav. So we'll start, as I said in the very beginning, our theme today is discussing QSP. And I personally also know very little about it apart from the acronym. And for most of our audiences, drug development, okay, we know we take the pills, and then when imagine drug development, we imagine white coated people running around the lab holding a pipette, looking under microscope, injecting a drug on a helpless mice, or maybe doing some clinical trial. We have some other interviews earlier where people worked on genomics or biostatistics in that sense. So we know statistics are used for clinical research also. But I believe quantitative systems pharmacology is a bit different from all these things, right? So can you please start with some simple examples that, okay, you are making quantitative models for pharmacology. What sort of model are those?
Absolutely. Well, first of all, it's a pleasure to be here, and it's very nice to be speaking to you after a very long time. To answer your question, I think some of it was in your introduction. So QSP, once again, for the listeners, Quantitative Systems Pharmacology. And if you pay attention to each term, all of it means something. The first one is quantitative. So the contrast from people, who are working on the bench in the lab, I think that's where this comes in, where, the experiments are quantitative. But what we are trying to do here is understand not just quantitative in a static sense, but how the system operates dynamically. So that is sort of implicit there. The second word is systems, right? So we're trying to understand how the whole system works together as a whole. And for those who are familiar with the idea of systems biology, this is very similar. um, we are trying to look at not just individual components, but how these components come together and how they influence each other. And the last part is pharmacology. So if you've heard the term systems biology, this is essentially application of systems biology to pharmacology, which is pharmacology essentially wants to understand what is a drug doing to your body and what is your body doing to the drug. And sort of that helps with the process of developing drugs. So if I to make these ideas a little more concrete, let's start with sort of what is your body doing to the drug. When you take a drug, it goes inside you, your body absorbs it, it goes into your blood, goes into different organs. So the study of this particular aspect, what your body does to the drug, is known as pharmacokinetics, or PK for short. So one key aspect of the modeling that we do is we try to quantify the dynamics of PK or pharmacokinetics. So we write mathematical equations, often in the form of ordinary differential equations, that are trying to, in a way, mimic what is happening to the drug as it goes into your system. So we might describe body in a simplified way as a set of compartments. So your blood could be one compartment, your organ could be another compartment, and you could make these models increasingly more complex. So that's one aspect of it. What is the body doing to the drug? But maybe in some ways, at least equally as important, or maybe more important, is what is the drug doing to your body, right? So once you understand how the drug is distributing in your body, you want to know what is it affecting, and is it affecting you in a way that is desirable, as in, is it doing what you want it to do? So that is where the biology and sort of the systems part again comes in, where you want to understand this drug is in my body, but is it going and binding to my target? How well is it binding? If there are, let's say, 100 units of the target, does it go and bind to all of them or a very small fraction of them? And once it binds, what happens downstream? So if it is binding to a receptor, for example, that receptor might change the behavior of how the system works. So we are trying to describe all of these things in mathematical terms, and in a sense trying to create a simulacrum of your body and how it interacts with the drug, and that sort of eventually leads to what we call systems pharmacology. And then you can use sort of these models to do a number of simulations, akin to what you might have done in a lab or in a clinical trial, where you can use these models to simulate what would happen if I take this much drug, if I take the same amount of drug every day versus every week. And you start thinking of questions like that.
So you are working in this field for long, almost a decade. So can you give us some example, like case studies type example that we teach in case of business schools, right? That in some example that you have seen in your life, or you have seen from heard from your colleagues, that these type of quantitative models, like suppose your pharmacology, pharmacokinetic or pharmacodynamic models that you have people building, has actually helped the company to make some critical decisions in the drug development pipeline.
Yes, yes. I think I will talk about an example that's in the public domain, um, and part of the reason for
Yeah, yeah,
doing
I
that,
understand.
right. There are some proprietary things that you don't want, but this is a very important example. In many ways, the model that I'm talking about is seen as the poster child of QSP, in the industry at least. So there is a disease known as hypoparathyroidism. There is a hormone called parathyroid hormone, which is secreted by the parathyroid gland, which sits next to the thyroid gland. And sometimes, very often it is because of thyroid surgeries that the parathyroid gland gets damaged. But there could be other reasons as well. So the patient does not secrete enough parathyroid hormone. um, and parathyroid hormone is very important for calcium homeostasis in the body. So if you don't have enough of this hormone, even though you're taking enough calcium by through diet, it will not get absorbed into your system. So just the basic biology of that is you need the parathyroid hormone, um, along with calcium as well as vitamin D. So you have to have all of these three things for your body to sufficiently absorb calcium. And calcium, as we know, it's very important for many things. Now, bones are obviously one of them, but it also is important for a lot of signaling in the cells, and maintaining sort of charge balances across cells. So lack of calcium has in your system has very serious consequences. So the standard of care for this disease used to be that, well, actually it still is that you take a lot of supplementary calcium. So you just take these sort of calcium tablets along with a lot of vitamin D. But the trouble is that your body is not absorbing enough calcium because you don't have this hormone. So by dumping a lot of calcium, you get more, just by simple mass action kinetics. Um, but then a lot of the calcium that you're eating is just going out into the urine, is being excreted out
Yeah.
through your kidney. And that is not good for your kidney, right? So hopefully this sort of tells you the systems aspect. This is why you want to understand how the whole system.
Everything is connected to everything. You try to fix here, something goes wrong there.
Exactly. So all of this is very important. So anyway, so there was and to make matters more complex, this is a rare disease. If I remember correctly, it might be an ultra rare disease. So the challenge with rare diseases is that even though you can conduct a trial, you don't often have enough patients to get enough statistical power. So again, for people who remember potentially from previous talks about statistics, it forms a very critical part of clinical trials. When you do a trial, you want to establish with statistical confidence that the drug is better than placebo. Now, in rare diseases, that's very hard because you have very few patients, so statistical power cannot be obtained. So this is one space where models of the kind that we work in this space can be extremely helpful, where you have limited data from patients, but then you use models to supplement their data. So there was a model built in, I believe, in 2010, of calcium homeostasis in humans, which very nicely described the whole system of PTH secretion, how it affects calcium homeostasis, and so on. And this model had been around, and people had been using it. So this is something that I have worked on partly, so that's why I'm sort of more familiar with this. Um, a drug was approved for this treatment as an adjunct to calcium supplementation. So you still take calcium, but the drug is a synthetic version of the hormone. So you inject it subcutaneously. You get more of this drug in your system, and it helps with the calcium absorption. Um, and the clinical trial that was done tested this drug at a dosage of once per day. So you take the drug once per day. So you're sort of, you're injecting it subcutaneously, gets absorbed, the concentration in your blood goes up, and it gradually dissipates. Um, so the FDA was approved this drug, but then they also decided to do some simulations of alternative dosing regimens using that model that was published a while ago.
In place of patients, actually.
In place of actual patients, exactly. The actual clinical trial was fairly small, um, on which this was approved, which is because, again, because it's a rare disease. So they did these simulations and tested out a bunch of scenarios. And what they found is that with once daily dosing, calcium homeostasis can be maintained for part of the day, but not for the whole day. And it might be better to, test the dose the patient maybe twice or more every day. Now this is again the sort of the practical side of it also comes in now. If you're asking the patient to inject themselves twice a day, it's challenging for them. So you also have to
Yes.
balance, right? So hopefully this is helping you see some of the nuances of drug development. um, so there is always this tension between what is most efficacious, but also what is safe, and also what can be practically done, right? So sort of companies are having to balance these things all the time, to make a commercially viable product. So eventually, the FDA asked the company that was marketing this drug to actually conduct more clinical trials with additional dosing regimens to see which dosing regimen is the optimal. And the model was sort of suggesting that twice daily might be optimal. So I think this is one of the cases where QSP modeling had a real impact, where the company was given an approval, but it was conditional on doing these trials to go and test whether whether the drug actually works. Now, if astute listeners can find this, I haven't named the drug, but they can find it and then sort of trace the history of it. The trial eventually did not happen, and then that drug had to go off the market for other operational reasons.
Okay. Okay.
It was a very good drug. It's unfortunate for the patients, but I think the upside of this was that the future generations of drugs that were developed, I think this was a key point, that you want a drug that remains in the system for a longer period of time, so that you don't have to dose as frequently, but you still get efficacy that is good enough. So now there is the next generation of drugs that has a PK profile that is longer, where you now don't have to dose twice. And the same model tells us that with that kind of PK profile, you don't need multiple doses within a day. So I think this is a very good example. The model was developed by authors called Peterson and Riggs, published in 2010, calcium homeostasis. So it's worth taking a look at this
Yeah,
model
it's
to.
very interesting. I should look into that actually. Possibly it
Yeah,
could be a case study for the students also to learn, right? Build the model and play with that, right? Yeah. That's very interesting. So one important interesting point that I find it, and it's very encouraging in that way, is that means you are saying that now regulatory authorities are now to some extent happy to accept mathematical models,
absolutely.
right?
That is
That's
true.
one major thing, because as long as they do not accept your model, you may make a very beautiful model which has very large power to make a prediction and everything. We are happy, but sometimes these institutions has their own momentum. They don't take decisions so early. They have to be convinced. But it seems they are moving in the right direction, right?
Absolutely, they are, and I think this has been, at least from the industry standpoint, this is a key thing for the companies. Is how do the regulators see this? We can use these models internally to drive our decision making, which companies do all the time. Um, but eventually, I think the maximum value for a company is when you can use these models and take them to the regulators and say, look, we have clinical data, but we also have supporting evidence, and the model might be filling in some gaps that we could not with the clinical or the preclinical or the animal data. And I think the regulators are sort of increasingly accepting of this. That space is still evolving. I think there are questions that need to be addressed, and there are legitimate reasons why the regulators have been hesitant up until now. And I think some of those questions have not really been answered, that the field needs to work on. And I think we can sort of get into that more specifically, if you would
So,
like.
so if I said these models, yeah, particularly this one, or suppose this ADME model, which is classical for
Yes.
pharmacokinetics model. So these are primarily sort of law mass action, ODE based type models, right? And then what you people do? Do you people do lots of parameter estimation, fitting the model with some clinical or animal experiment data?
That's right. Yeah. And I've been talking about sort of, I've been limiting myself to the QSP aspect. But as you said, so there's a whole other world of sort of smaller models, which are not as mechanistic. I think you can think of them as semi-mechanistic or maybe sort of more abstract models in the ADME space, ADME space.
Mm-hmm.
And for the listeners, ADME is absorption, distribution, metabolism, and excretion, essentially what happens to the drug once it goes into your system. So these are models that are often not truly mechanistic, but somehow describe and capture the dynamics. That space has come to be known as pharmacometrics, and it is older and more familiar to the pharma industry than QSP. I think QSP is relatively new. So pharmacometrics, the approaches that modeling takes, they focus on building simpler models. So for example, we were talking about pharmacokinetics, what the body does to the drug. You might choose to describe the whole PK of a drug by treating the whole body as just one compartment, and that might work for some drugs. You say, the whole body is
Yeah.
just one blood and goes in, goes out, and that kept. Sometimes you take two compartment's, and that's enough. Even though the body is very complex, you've abstracted it to just two little boxes, and that often works. So by having these smaller models, parameter estimation becomes easier. Those parameters may not have real meaning, right? They do not translate one to one physiologically
But
to the
have
body.
predictive power.
They have plenty of predictive power. And the second thing that happens is that you can marry that up with statistical approaches to get statistical estimates of how confident you are in the parameter and what is your error in estimate. So,
Mhm.
in pharmacokinetics modeling, when we do these models, we actually not just provide the parameter, but we also say what is my relative standard error for each
Confidence
parameter.
level.
Exactly. So that they gives me my confidence level. So you are able to verify, capture the variability in the population while capturing the uncertainty in your parameter estimates. So those sets of models or that set of models, because it has sort of a stronger statistical backing behind it, are more palatable to everybody, right? So you have
Yeah.
more confidence in what you're doing. Um, and I think that's also partly driven by, so I mentioned simplicity of the models, but also those models tend to have more data. PK is often quantified very nicely, and so that becomes easier. But when you go to QSP, you're trying to capture sort of the dynamics of a very big system, so estimating parameters become very hard.
Yeah.
And we can't do the things that we do in pharmacokinetics. You could try applying those same mathematical techniques, of course, but and probably not going to work. So the approach becomes different. But again, going back to your original question, a lot of what both of these spaces are doing, pharmacometrics and QSP, are very similar. We are, I believe, 95 to 99% of our models are ODE based models. We were writing differential equations to describe these things. Sometimes we'll do partial differential equations. When we are trying to be really fancy, we might look at agent based modeling and things like that.
So agent-based modeling also comes into this space.
Yes, agent based modeling is
Okay.
something that people look into for specific questions where sort of the spatial geometry becomes
Yeah.
important, or
So that is
interaction
much easier to do
between.
than PDE.
Correct. Or sometimes you're trying to look at interactions between different kinds of cells. Again, agent based models sort of are better suited for that. Um, and then sort of parameter estimation is a key part of this.
Yeah.
So you have to figure out optimization algorithms to estimate parameters. You might need optimization algorithms to figure out what is an optimal dosing, what is a good sampling schedule to best fit your parameters. And I think the other aspect of QSP that's very important is creating virtual populations. So you can create one instance of the model that say captures your average or your typical patient, but not everyone is the same. So what we do then is we say, okay, we fit the model to the average. Now we'll identify some parameters in the model that the output is most sensitive to, and let's vary this over some space and
So you
train.
randomize that parameter.
Yes,
You do
randomize that.
random sampling from a parameter space.
Exactly. Yes. And you
Okay.
create a lot of so-called virtual patients to get
Right.
a virtual population. So you can treat all of these patients in a population. So it's kind of like a large clinical trial, right?
Yeah.
And
Yeah.
then when
Yeah.
you have
Yeah.
this big population, you can sort of select certain individuals, analyze them in different ways, and see who's responding, who's not responding, why, and so on and so forth.
Yeah, it brings to a point that actually I keep on telling many a time when young researcher, PhD student, and some people who are now it has become a fashion. There are also that we say that there should be one model at the end of your publication, right? In biology, that is becoming like that. And so I sometimes get a question from young researcher that where does the modeling stands in my work? So I say that if you are doing experimental biology and if your experiment can reliably give the answer, then you don't need the model. Then you are not doing, I mean, it is worthless. Because in science, ultimately, hard evidence is, you are not doing math, you are doing, right? But there are questions which you may not have the technology, you may not have the resources, which you can answer by your direct experiments, right? It can be technological challenge, it can be monetary challenge also sometimes to arrange the things, right? So in that case, your model can actually useful from their
Yes.
perspective, right? So that's what you are actually doing. Where a real clinical trial or the real animal test I cannot do. Let put it support it by experiment or reduce the space where I have to do the experiments.
Yes.
Yeah,
Absolutely.
that's what I get. So nowadays in everything people are trying to incorporate genomic, proteomic, this type of data. So in QSP also people you try to pull those information in your model somehow.
Um, Yes, sometimes. So I think it might be relevant in the context of virtual populations that we were talking about. So, for
Mhm.
example, if you have a disease that has some kind of genomic variability
Mhm.
that leads to different kinds of patient populations,
Uh-huh. Mhm.
right? Eventually, I think because QSP modeling is very phenotype driven in that sense, right? So
Yes,
you sort of
yes.
want, right? So you want to connect the genotype with the phenotype. So if the genotype is indicative of variability in the phenotype, we want to capture that. And this reminds me of one specific example in IBD, inflammatory bowel disease,
Mhm.
where I was working with a group where we were looking at kinase inhibitors, a particular type of kinase inhibitor to reduce inflammation in the disease. And it turned out that the particular kinase that this drug was targeting, there were individuals with who were sort of um, just genetic variants who had naturally less sensitivity, were less sensitive to this particular kinase. So the drug might not work as well. So I think those types of information that we get from genomic studies are applicable here. And similarly from proteomics, I think proteomics is more important because that sort of directly fits into a lot of modeling work that we do. Where if we know sort of what is the let's say expression levels of a
A
protein.
particular enzyme or a receptor, something like that.
Exactly. So those can be captured in our models. But I think what is key to our modeling is understanding the mechanisms of how things
Mhm.
connect with each other, how the
Mhm.
whole system operates. So
Mhm.
for people who are not sort of familiar with the field, an essential aspect of any QSP model, and you will see this in pretty much every paper, is is a diagram that's describing the system, somewhat trying to mimic how engineers do it, and trying to sort of link things with each other. And which reminds me of a very nice paper. And I'm sure you're familiar with this. It's called "Can a Biologist Fix a Radio?"
Yeah,
Um,
that's that. In my systems biology class, I always start with that. Yeah, yeah.
yes,
I think that everybody should read. That everybody
yes,
should read. Yes,
yes.
everybody should read that. Yeah, yeah. So just a fancy question because everybody talking about it nowadays. So in every field, people are trying to use AI/ML. What about that? Is there any scope or something?
Yes, yes. I think people are very actively thinking about this in this space. And I mean, when I think about it, I think there are maybe broadly two or three categories that this falls under. The first one is, because I think AI has caught people's imagination after the advent of LLMs and these chatbots. So I think one of the things that people are trying to do now, and I think some of the agentic LLMs are already doing this, they're doing it quite well, is when you're trying to build a model, it takes a lot of research. Read a lot of papers, try to understand the mechanisms, what interacts with what, and then you sort of make this sort of a schematic of conceptually affects B, B
Yeah.
affects C, and the next step is finding out how strong are these connections. If A
Right.
affects B, how strongly, and all of this primarily for people like me, who are not sort of doing experiments to gather information about or data about everything. We go to the literature and look at what's known. So I think that's one natural space where LLMs fit in, where they give it a problem, it can go and do a web search and find all these papers and I've actually done this just experimentally with one of the products that are available. Um, and I asked it to make a model of, I was just experimenting, and I said, "Can you make me a model of blood coagulation and include the key components, make it as comprehensive as possible, but try to keep it within less than 50 state variables?" And it went and did its research for about 20 minutes and came up with this beautiful report, very nice
Yeah.
diagrams, right? Right. And it would have taken me weeks of research to get that information.
That's
Now all I
true.
can
That's
do
true. And
is
manpower also, not just you. You may have to take help from
exactly,
somebody else. Yeah.
exactly.
Yeah.
So I think that's one space that kind of naturally fits, and people are now working on. I think as recently as last month, there was a paper that talked about developing sort of chat bots that you can give papers to them,
Yeah.
and they will build these sorts of networks.
Mhm. In fact, your Claude and other has already started LLMs which are specific for biomedical
Oh wow!
space. Actually. Yeah.
Okay, I should look. I did not know this. I'll have to look
Mhm.
that up.
Yeah.
Um, that's one space, and I think the second one is I think sort of aside from LLMs, there's a number of other machine learning techniques that people have been using. These are sort of bespoke models that answer specific questions,
Mhm,
uh,
mhm.
and I think not just in biology but also in the industry, even in tech industry and other industries in general. I feel like those continue to be the workhorses where they're actually doing the actual work despite all the excitement around LLMs. So that's the other space where I think um, people have been for a while now combining QSP with machine learning methods to enhance the power of QSP models in a way. And a little piece of work that I did with some colleagues, one of whom is Sai, also a former student. So we published a paper a couple of years ago on modeling of Crohn's disease, where we combined machine learning with QSP modeling. And the machine learning part was looking at some patient features, their baseline features, and using those features to predict whether this patient will is likely to respond very well to a treatment or sort of not so well or not at all. So it puts the patient in one of these three archetypes, and then with these archetypes, when we go to our QSP part, we can parameterize our model in a way that sort of fits these archetypes and then do the further work.
Mhm,
So it
mhm,
sort of
mhm.
enhances what QSP can do on its own. And I think there are many other efforts like that, which are using QSP along with machine learning to answer specific questions that might not be answerable with QSP alone. And then the third aspect that I'm seeing is also developing is the so-called physics-informed neural networks. So
Yeah.
neural networks, right? So people are trying to sort of build in mechanisms of biological systems into the neural networks, so
Put
that
some constraint based on biology and physics.
exactly. So that sort of becomes another way of capturing the behavior of the system in a way that's constrained by the mechanisms that we want them to capture. Because otherwise, neural networks will fit the
Yeah,
data
it fit. Yeah.
exactly, but as soon as you
And
try,
your data is sparse actually. You are not lucky like
exactly,
Amazon customer
yeah,
data set, which has trillions of data point. You have a limited data set. So neural network will go in every direction. Right?
yes,
Yeah.
exactly. So then predicting out a sample with neural networks and predicting, for example, new dosing regimens, they just collapse. Whereas
Mhm. Mhm.
physics informed nets
In
are
fact, in fact, long back, actually, one of my friend, actually, I was arguing that pharma industry in general is not very hopeful about AI, particularly large neural network and all these things, because biology inherently till now is data sparse. I cannot do a experiment with the 1 million animal. Forget about human, right? So I cannot have that amount of data. So these large models which require huge data are not suitable in that sense.
absolutely, that's correct. And but the space where I see actually these kinds of models working out, especially knowledge graphs, is um, sort of gathering information from literature and sort of automating a lot of this thing about how are things connected, and creating sort of annotated information. So the likes of KEGG, for example,
Yeah.
the Kyoto Encyclopedia of Genomics,
Mhm.
but a lot more um,
Automatically generated and
correct, correct. And I suppose the challenge with those approaches is, again, how much do you trust AI? Because someone will have to go in and manually
yeah,
curate it some.
that's true. But anyway, still that reduces the workload, the manpower you require, and the time you require. So if I come back, see, to your experience of this whole almost 15 years in this industry, see, you when you look from the top and you are seeing these success and failures, so I can put this area broadly under the category of mathematical modeling of biology, right? So what do you think the most friction, most limiting situations, things in that?
Yeah. And I think challenges are slightly different depending on sort of which aspect. So I'll focus on QSP again for this discussion, which is sort of these sort of relatively large system type models. And I think we talked about some of this before. I think one of the big challenges in this field is, how do you quantify your confidence in the model? I think that's a huge challenge for us. Unlike the pharmacometrics models that I was talking about, we still don't have a singular measure of accuracy. So this is something that I have dealt with, pretty much all my modeling career, is when I build a model and I go to stakeholders who are not modelers, who are ultimately the people you're trying to influence. These could be clinicians, these could be regulators, these could be biologists you're working with. Like, how accurate is your model? And I don't have a numerical answer to this question, so I
Yeah.
have to sort of beat around the bush and be like, this part is correct, this is uncertain. So I think until we come up with a good way of answering that question, I think we will continue to face a big challenge here. Or we come up with some kind of vocabulary so that it's easy for us to explain that these are the limitations of the model. But I think the challenge with that is, that's a very verbose answer, and nobody has the time
Mhm.
to,
Yeah.
understand. So I think that is a fundamental challenge for this space. How do we
Okay.
get to a point where we can have a reasonable estimate of, this is a 50 equation model and 75% reliable, right? I think
Okay. So
something,
some sort of measurement, some sort of metric.
yes,
Uh,
exactly. So that's one, I think. And then the other one is, we promise to represent systems, but anybody who knows these systems really knows that there is no hope of truly capturing the
yeah,
system.
it is still a subsystem.
Exactly. It's a subsystem, and even within those subsystems, there's lots of abstractions, right? So you're
Right.
simply
That's right.
handling,
That's right.
things. Um, so I think a lot of us in the field have come to look at this as, which I think, in my view, is a reasonable view, is you're not trying to capture the biology, but you're trying to create a model that has some predictive utility, right? That
That's
is your
true.
ultimate goal, right?
Hmm.
So there is this sort of trade-off between complexity and predictability. And again, what happens is when you talk to people who are not specifically from this field, I think the question is, oh, well, I know about this particular interaction that's not in your model. So how can your model be correct? Right. So then you
Hmm.
sort of get into this debate about which are the important interactions, why is my abstraction
Keep and
right?
what to throw.
Exactly. So I think that becomes another challenge where
I think that is actually, a sort of philosophical position that comes through training, because
Mm,
when you start looking from a physical perspective, more mathematical perspective, you start believing that the truth is in the simple thing.
mm, mm.
You want to focus
Mm.
on the forest rather than counting every
That's
tree.
right.
So
That's true.
that comes from your training in that field, whereas in
Yes.
certain other field where they do not get trained in that philosophy.
Mm,
See,
mm.
nobody has trained us explicitly in a philosophy class, but
Sure.
the way
Sure.
you learn physics, engineering,
Yes.
and math, that is the implicit thing. Make it
Absolutely.
simple, but still working,
Yes.
right? Whereas in certain other field, people get trained from the very early days that you have to
Yes.
look into the detail, fine tune everything. So
Yes.
when
Yes.
you talk to these people, particularly biologists, because they are coming from that philosophical position,
Yes.
it
Yes.
becomes a problem because the initial positions, belief systems are completely different. We are
True.
talking
That's so
about,
true.
right?
That is so true.
Yeah,
Yeah,
that,
absolutely. Yeah. Yeah. And I think, I mean, the sort of the two things we talked about, the accuracy quantification and this complexity versus predictability trade-off, I think these things are not answered really well yet, which then really leads to the practical challenges where people who really matter, the clinicians who are running the trials, whom you need to convince that this is the right dose, for instance, or the regulators, they don't accept it as easily as one would hope. So
yeah.
I think that becomes a big challenge. And I think the other aspect you already sort of talked about it is the data aspect. These are big models; they need a lot of data, but we are by default, data sparse,
Data
and the
sparse.
data when they're available are super messy. The error bars are bigger than
Yeah.
so sort of how do you deal with all of this? And I think from where I stand, I think the other aspect is also finding the right talent who sort of appreciates all of this, and in a sense is bilingual, at least bilingual. They understand the biology and the math, but then they have the
Yes, yes,
computational
yes.
prowess to follow this together. So that's not. I think there is sort of universities are training more and more of these people, and there is more interest in this. Um, but I think we are sort of still on an upward curve, but we're not quite where we have enough people to, naturally fill all of these spots. And I think AI, with its sort of as it has come in, has sucked up a lot of this talent into it. So I think that
Okay,
will
yeah.
continue.
Yeah. That brings me to this question of this talent. See, you must be hiring people, you must be building teams over these years, right? So when you pick up your colleagues and you build the team for this type of problem, right? What sort of academic background you are looking in them?
Yes, yes. Yeah. So I think this is an interesting question, and I've written some job descriptions where sort of, I want to capture the degrees that I want to accept, and that list is very, very long. But I think the summary of that is as long as you have a quantitative background, um, where you understand, I would say, college level math.
Hmm.
I think that is great. That is what we are looking for. And if you have some appreciation for biology,
Hmm.
so graduates from maths and physics, from lots of engineers, some people who are even coming from computational sciences, obviously pharmacology and physiology graduates who have done modeling work. Like you said, some of them are doing doing biology or pharmacology, but they want to have the model at the end of the paper, so they understand the space. So I think all of these are welcome, and I think from the engineering space, I think bioengineers are naturally trained to do some of this, but I've been particularly impressed with chemical engineers. I think they have a
Yeah,
very
because see, your ADME model is nothing but
yes,
chemical engineering models, actually.
exactly. So they have a very sort of intuitive feel for these kinds of models.
Mm-hmm.
Um, and so do I think so do a lot of physicists. One of the smartest QSP modelers I have had the opportunity to work with was a physicist who, after his PhD, chose to go into biology. So did a postdoc in a bioengineering lab, and published some really beautiful models of cell signaling. So all of these spaces are good, and I think the biology aspect, as long as you appreciate how biological systems work, you don't have to be a very deep biologist, because what often happens with modelers is, unlike biologists who are specializing in one aspect or domain of biology, we are expected, at least on the industry side, we are expected to apply these skills across a number of. I have worked in
Domains.
metabolic disease. Exactly. I've worked in metabolic disease. I've worked in inflammatory disease, bowel disease, rare disease. Now I'm working in oncology. So these principles will apply across domains, and you will always have people to help you with the biology and physiology side of things. So I think the quantitative side, having that systems thinking and being able to appreciate, how biology works, and being able to read people papers and sort of quickly come to um, terms with sort of how the system work. I think that is the critical skill.
Yeah, yeah, maybe to develop the ear that so that he can understand what the biologist is saying.
Absolutely, absolutely, yeah. And I mean, I think that's where sort of the bilingual aspect comes in.
Yeah, that's
And I
right. I
know
agree
that,
with that.
right, you need I think you need to be more conversant in math and be able to understand biology, even if you're not able to speak it fluently, but at least understand it and describe it in terms of math. um, and I mean, this sort of vocabulary of speaking in multiple languages is something that I know that there are some bioengineering programs who that actually are sort of actively trying to bring this into their curriculum. And I went to one of them, in my PhD program, and I worked with you, and I know sort of you also sort of think that way. And again, I'll take this opportunity to thank you, because I think sort of my interest in QSP and systems modeling, came a lot from the work that I did with you as an undergraduate. So I think those are the skills that sort of people need, and of course, for all of this to work together, you have to have computational skills, to put this to work in a computer. AI can help you a lot with that these days, but still you need to understand the basics. And I think the thing I've seen sort of both from the academic side and based on the last question that we were discussing, after having done all of this, being able to communicate this in a way that is palatable to different kinds of audience, I think that maybe in some ways is the harder skill.
Yeah.
And unfortunately, our engineering and science programs maybe don't prepare us for
Yeah,
that
yeah,
very
yeah.
well.
You're right. You're right. I think I keep on fighting with my BTech, MTech, PhD student to convince that, see, your PPT, your report, or when you are showing me the data, this should not be for you. It should be for me so that I can understand, right? Yeah, that's true. We are not teaching these kids the importance of communication and how to communicate actually. Yeah, that's right. So, from your last description, actually quite encouraging for me, because some many a time I find these queries even in the campus. Somebody studying engineering physics, somebody in computer science, or suppose we have in data science school, even electronics, and some couple of mechanical engineering students also worked with me. So they have this knack that because they read popular science articles, they know that biology is almost like the new space challenge for us, right? So they are excited, and they sometimes get excited from the 101 course, but they are very apprehensive that, okay, I have never studied biology. Will I be able to jumpstart into this area, right? So I always tell them that, okay, computational mathematical biology is the right place, or suppose physical biology, those are the best place to go for you. So what will be your suggestion or advice for them?
Yeah, yeah, and so 101 is very interesting. I don't know if it's still the same, but 101 was also
Okay,
the thing
it
that
changes
really
with
got.
the teacher, but the subject is same.
Yes. Okay. Yeah. Yeah. That was also the thing that actually got me excited, and I think then I decided to make this because so when I joined IIT, I was not in biotech or bioengineering. I was in different branch, and I changed my branch into bioengineering because of. And I think a lot of us who come to engineering, we are interested naturally. I think in sort of this systems view, whether we realize it or not. I think that's sort of something that draws us there. Apart from sort of job opportunities, perhaps. Um, but I think that 101 course to me, sort of made it very concrete to me that biological systems do operate a lot like very well orchestrated machines, and there should be ways to describe it. So I think that's how I. So if that kind of thinking is driving you, I think you're already in the right place. You're halfway there. And then I think it's the rest of it is a question of building out some of the skills that we talked about. And I think as we were talking about, the previous question, hopefully it was evident that this is not a one man sport, right? This is very much a team sport. So you can come into that team sport from many different angles, right? If you want to be a biologist, that's still very good. You could work with people who are more quantitative and sort of help them understand the biology. And this is something that we routinely do, because as long as I've been working in this field, I think there's lots of things that I don't know about biology. For example, sometimes questions as simple as, I have this binding affinity measured by two experiments. Which one should I use for my modeling? And I don't know the
Right.
techniques as well as a biologist might,
Mhm,
right?
mhm.
Which is more applicable to my animal or human model? So I think there's that. But I think for people who are coming from a more quantitative background, I would say the fact that you have the interest is a great start. I would say strengthen your computational core. I think that's very important. You have to have a good understanding of ODEs, differential equations, numerical methods, linear algebra. These things are important.
Probability theory.
Probability, absolutely. And then try to maybe pick up on some aspect of biological domain. So to me, for example, I mean, I was a biotech student, so I studied molecular biology and all of this, but immunology really appealed to me, right? I felt like that has a really nice
Mhm.
uh, ring of this sort of systems where such a complex system with so many
Multiple
players.
things come together to win the race.
So maybe sort of look at some of these things at a surface level, maybe at 101, and you will see there are some things that appeal to you. And maybe sort of try to delve deeper into that so that you feel comfortable with biology. For people who are coming from the physical sciences, I think one thing that really is different in biology is that the data are never as clean. These are super messy data. And I have worked with statisticians and physicists and engineers who come here and look at my models, and they're like, wait, what how can you model this data?
That's true.
right?
Particularly physics people who does not want more than one parameter in the model. Right, you have 57 ODEs with maybe 2 3 into 57 parameters for which you don't know
Absolutely.
values.
Absolutely. So I think that, that sort of, that takes a mindset shift. For me, it was the opposite because I was coming from biology. I had done sort of biotech and I've looked at biological data all my life. So when I first look at, I remember sort of in my undergraduate days, when I first look at climate change data, I was like, wait, this pretty much looks like a flat line to me because, from a biology perspective, this noise, it's, so it sort of, that helped me understand. So maybe sort of look at these data sets and see, the scale of variability and uncertainty that you have in these measurements. That will help you think about things as well. And I would say, as we were talking about maybe picking up some project, doing it hands on. And for people who want to start really light, I would say maybe start with PK models. These are the simplest models. Look at pharmacokinetics models, 2, 3 compartment, and sort of build on the complexity. And work on your coding skills. You pick up one language. It could be Python's becoming increasingly popular. Pharma likes to use R a lot because it's also open source and it's preferred by statisticians. MATLAB is one. Learn about QSP modeling is, it's fairly complex. And this is something that I sort of picked up after my PhD. I wish I knew this in my PhD. Learn about version control and some software development principles, because your models can very quickly become very complex. And,
The
knowing
project can become trunky very easily.
exactly. So you need to have some sense of sort of how to write good software. You don't have to be a software developer, which again, this is also a pitch to software developers. We need more software developers to come in this space and, teach us those principles and force us to write good models, not just hacky code that runs and, does the optimization. So these are other things that, some may be natural at, but then they can build their skills in the other spaces. And then I think from the standpoint of sort of entering the industry, I feel like sort of, going to graduate school, etc., is the natural course. I think if you want to be a scientist in the QSP space, you need at least a master's degree, preferably a PhD. So I think that's the path. All the scientists that I work with, everybody has a PhD. Um, you may be able to get away with a master's if you're not sure about whether you want to go into a PhD. I think master's is a good choice. Go and do it,
Right.
right,
Yeah,
intermediate
you can
space.
branch out depending upon at that time,
Exactly.
because it
Exactly.
is very difficult in early to decide what you want to do in
Yes.
life.
Yes, yes, and I think internships, if you can find them, are a good way to learn about this space. I did internships both as an undergraduate in an academic lab. So both in my second year and my third year, I went to academic labs in India, or I also had an opportunity to go to a foreign university to see what these labs are doing. And I mean, I think I kind of had a sense that I wanted to do quantitative biology early, so it was easy for me. I could pick those labs. And also as a PhD student, that was my first brush with industry. So I went and did an internship at a major pharmaceutical company, where I first learned about clinical pharmacology, and what is clinical pharmacology and what does pharmacology mean. And I had the opportunity to build one of the first QSP models in the IBD space, which was published. So there are ways that you can sort of learn about what is happening either in the academic space or the industry, and that helps you understand the space, but also builds the connections that you can then use later on
Yeah.
as you want to
Yes.
grow further.
Yes. That's right.
And I think also I think the other advice I have for students is, now that you have LinkedIn, reach out to people in the space that alumni and so on. And I've had the chance to talk to students here locally in Boston. They network with me and others in the space and try to sort of just do informational interviews. Feel free to reach out. Worst case scenario, people will say no. Sometimes when they have time, they will say yes, and you'll get a chance to talk to them and learn more.
You're
Um,
not losing anything. You're
yes,
gaining actually. Just right.
exactly. So that's another way to sort of learn about the space and build your network.
Yeah, I think you have broadly covered almost all the aspect that one should keep in mind when planning that, how they should move forward in general. But still, do you have any specific advice for people, suppose from our department or somewhere who they are doing BTech in bioengineering on biotechnology? Anything unique apart from these general advices?
Yeah, I think for the students in bioengineering, I think these are probably more privileged, in the sense that they already have a lot of these ingredients, whether they appreciate it yet or not. Because I think these sort of, the molecular biology, the genetics, the lab work that we do, I would just say, sort of try to appreciate those things a little more right now. You perhaps don't know yet that you want to go into quantitative biology. So keep an open mind to all of these courses and don't treat, because I think, when I was an undergraduate, a lot of us, when we had to go into the lab, who knew we were going to do mathematical work, we sort of really didn't have a lot of respect for that work. But now I know, that it's very important to understand sort of how these data are gathered. um, not just from the standpoint that, it's a lot of hard work, but when you actually taking that data and putting them into your model, they influence your model in specific ways. You need to know how they were gathered and sort of how do you fit the data and how do you interpret the data. So having that appreciation is important. And I think the other aspect I would encourage is, I think for some of us, coding was a natural thing. For others, it was not. But I think either way, you need to be good at writing code. And I think a lot of this is stuff that you don't appreciate until you are dealing with practical challenges. So maybe,
Right.
challenge yourself with picking up a project, or do a series of projects on your, maybe even on your own, right, and of increasing complexity, and see why you need these things. I think that would be really useful. And also you could build a portfolio of projects, that you could use later on. This is something software engineers do frequently, where they sort of do their own projects and have a
Put
GitHub
a git.
page, right? So you could do something like that. And when you apply for grad school and so on, that could be a part of your application, or even for a job. It will come in very handy at the end. And again, as I said, try to look at practical, clinical data. You can find some data on the internet. You can find animal experiments, and again, reiterating, but build your network. I think these are
Yeah,
very
I think
important.
you have hammered the right point, building network, because as I see, most of the computational work can be done with a desktop and a good internet
Yes.
connection. So if somebody is very much inclined and desperately looking for someone to connect with, even now many clinicians are also interested in this type of thing. They
Yes.
also have nothing to lose, right? They can simply tell, okay, you do work on this, give me if you can create a model or something. But these students has to go and approach these people, clinician,
Yes.
companies, right? Somebody, or even some academician who may be doing experimental biology but want the model at the end of the paper.
Yes.
And PhD student cannot do that, but one of
Yes.
our BTech guy
Exactly.
can possibly do that. But we have to take the initiative from our side, right?
Absolutely. Yes.
That's
That
what.
is
So networking is key. We have to approach people.
yes. And I think the last thing, again, I will touch upon. This is, again, we mentioned in the last question, is the communication aspect.
Yeah.
Right, communicating technical information to a layer audience, and not as in they don't know science. I think sometimes the challenge is that they know too much science, right? So then, sort of convincing them that what you have done, sometimes it can be very hard. So work on those skills, and you get that, through presentations that you do, learning how to structure your presentation, how to communicate your ideas in a way that's simple to understand. Sometimes, as scientists, we get so enamored by all the fancy stuff that we've done. We want to talk about all the fancy stuff, and we sort of lose the message. So that's a hard thing to
That's right.
focus on.
That's right. That's right. In fact, that remind me today itself. I was this Julia Con is going on, Julia conference, right? So I was watching one lecture. So there, the fellow was saying in one slide, he was showing that it is said that your code has been written so that a human can understand always, but machine can interpret sometime. So you have to communicate to the human being, right?
Yeah,
Whether you are writing a program or you're writing a thesis,
that's
writing a paper or a presentation, that's communication is the key. Yeah, that's a nice point. Actually, miss that, and somehow we do not train it in an institutional way. That is unfortunate. It's true.
yes.
We do not train students in that way, in that sense, right? Okay, Gaurav, we had a long time, almost 1 hour. We are discussing. It was a very exciting, experience for me. that how simple OD based model that we teach to our school students and can become a real application in the industry, right? And thank you for joining me for this podcast. We are meeting almost like more than 15 years, so it's a very exciting thing in that sense also. Thank you for joining me.
No, I really enjoyed the conversation, and thanks for inviting me.
Thank you.